Hello, welcome to World of Microbes. In today's video, we will discuss forensic biology and its subdisciplines. Basic and optional field: molecular anthropology, forensic botany, forensic odontology, and forensic serology. Do like. In forensics, sample collection involves carefully securing a crime scene, identifying potential evidence, using appropriate tools to collect samples like blood, bodily fluids, fibers, fingerprints, or trace evidence, properly labeling and packaging each sample to maintain chain of custody, and documenting the collection to ensure the evidence's admissibility in court. Key steps in sample collection include: isolate the crime scene to prevent contamination and preserve evidence. Conduct a thorough visual examination to locate potential evidence like bloodstains, footprints, hairs, fibers, or trace material. Take detailed photographs. Create a sketch location of each piece of evidence. Wear gloves, gowns, masks, and eye protection to avoid contaminating the scene or evidence. Sample collection takes place through swabbing. Use sterile swabs to collect bodily fluids like blood, saliva, or semen from surfaces. Scraping. Scrape dried samples like blood or debris or razor blade onto a clean piece of paper. Tape lifting. Use adhesive tape to lift trace evidence like fibers or hairs from surfaces. Vacuuming. Vacuum loose fibers or debris using a specialized vacuum device. Fingerprinting. Develop latent fingerprints using powders, chemicals, or lasers. Packaging and labeling. Place each labeled container appropriate for the evidence type, e.g., blood in a sealed vial, fibers in a paper envelope. Document the collection process, including the collector's name, date, time, location, and case number on each label to maintain chain of custody. Store samples at the correct temperature and humidity to prevent consideration. Use new gloves for each sample. Avoid touching potential evidence with bare hands, and change swabs frequently to prevent cross-contamination. Follow established protocols for collecting specific types of evidence to ensure accuracy and admissibility in court. Once collected, samples are sent to a forensic laboratory for qualified experts to identify and compare the evidence. Do like and subscribe. Forensic science is the use of scientific methods to solve crimes and analyze evidence in court. It's a key part of the criminal justice system. The basic principle of forensic science is that every contact leaves a trace. This principle helps investigators link suspects to victims, objects, and crime scenes. Full-service crime laboratories offer services, including DNA analysis, forensic biology, and chemical analysis. Forensic biology includes. It also helps in criminal identification by creating facial composites from witness descriptions, craniofacial reconstructions, and preparing graphic presentations of evidence. Forensic scientists analyze scenes and in laboratory. They use a variety of techniques, including serological tests to identify body fluids, examining body fluids like blood, semen, and saliva, examining skeletal remains, hair, seeds, and other biological material, helping with rape and murder cases. Do like. A case receipt unit is the department or section within a forensic laboratory responsible for receiving, logging, and initially processing evidence submitted for analysis from law enforcement agencies or other requesting parties. Essentially, it's the first point of contact for new cases coming into the lab, where evidence is documented, assigned, and prepared for further examination by specialized units within the laboratory. Key functions of a case receipt unit: evidence intake, receiving and documenting all evidence submitted, including physical items, biological samples, and digital media. Chain of custody, maintaining a detailed record of evidence handling and transfer integrity throughout the analysis process. Case assignment, distributing evidence to the appropriate forensic discipline within the lab based on the nature of the case. Data entry, recording case details, including relevant information like the crime scene location, suspect details, and requesting agents. Preliminary examination, performing actions of evidence to identify potential issues or areas of focus for further analysis. Packaging and storage, properly storing evidence in secure containers to maintain its condition until further processing. Do like. In a forensic science laboratory, the biology division in the case receipt unit is responsible for receiving and managing evidence containing biological material, such as blood, semen, saliva, hair, tissue, or other bodily fluids, which will be further analyzed for DNA profiling or other biological examination of potential suspects in a criminal case. Besides, it also helps out the investigating agencies in crime cases involving rapes and murders on the spot. They handle evidence like blood stains on clothing, swabs from crime scenes, bodily fluids, hair samples, and sometimes even skeletal remains. The division receives the evidence, carefully examines it for potential contamination, and properly packages it for analysis in the biology lab. They maintain a strict chain of custody, documenting every step of evidence handling to ensure its integrity in court. They may communicate with investigating officers to clarify details about the evidence collected and its particular case. His main function is to extract DNA from the biological evidence and perform DNA profiling to compare against known samples, like a suspect's DNA. This division is crucial for cases involving sexual assault, homicide, robbery, and other crimes where biological evidence may be present. The serology division is the section responsible for receiving, examining, and analyzing biological evidence like blood, semen, saliva, and other bodily fluids to identify their presence and characteristics, often as a preliminary step before further DNA analysis is conducted. This division was created as a result of the effort of serological examination. Serology division primarily focuses on identifying the type of bodily fluid present on evidence items like clothing, weapons, or other surfaces. Responsible to find out the species of origin, blood group substances, enzyme, serum protein etc. in the biological materials such as blood, saliva, epithelial cells, tissues, bones, hairs, teeth cavity scrapings etc. Once a bodily fluid is identified by serology, the evidence may be sent to the DNA analysis section for further individual identification. Physical science unit in forensics refers to a specialized laboratory section within a forensic science facility that utilizes principles from physics and chemistry to analyze physical evidence from crime scenes, including things like trace evidence, fibers, paint chips, glass fragments, soil samples, and even materials from vehicle accidents. Identify potential connections between suspects and crime scene. Key aspects of a physical science unit in forensics include analysis of trace evidence, examining minute pieces of material like fibers, hair, paint chips, and debris to compare them to potential sources. Glass analysis, analyzing the composition and characteristics of glass fragments to a specific source, like a broken window at a crime scene. Soil analysis, studying soil samples to identify potential locations where a crime may have occurred based on unique soil property. Paint analysis, comparing paint samples from a suspect vehicle to paint chips found at a hit-and-run scene. Firearms analysis, examining specs and projectiles to link them to a specific firearm. Tool mark analysis, analyzing tool marks left on objects to match them to a specific tool used in a crime. Common techniques used in a physical science unit. Microscope, using microscopes to examine trace evidence for detailed characteristics. Spectroscope, analyzing the chemical composition using techniques like infrared spectroscopy, IR, or atomic absorption spectroscopy, AAS. Chromatography, separating and identifying components of a mixture, like analyzing ink sample. Elemental analysis, determining the elemental composition of materials using techniques like X-ray fluorescence, XI. Firearms unit forensics is the process of analyzing firearms and bullets left at a crime scene. The goal is to link the bullets to weapons and the weapons to people. How is this done? Gunshot residue analysis, a forensic technique that examines microscopic particles expelled from a firearm when it is discharged, allowing investigators if a person was likely near or handled a recently fired gun by detecting these particles on their skin, clothing, or other surfaces. Typically, this involves identifying the presence of elements like lead, barium, and antimony, which are key components of a firearms primer, using a scanning electron microscope, SEM, with energy dispersive X-ray spectroscopy, EDS, to analyze collected sample. Comparison microscopy examines two objects simultaneously using a comparison microscope, a specialized tool that allows investigators to simultaneously view and compare two separate samples side by side under a microscope, primarily used for analysis like fired bullets, cartridge cases, tool marks, fibers, or other items to determine if they originated from the same source, making it a crucial technique in forensic ballistics and other forensic disciplines requiring detailed comparisons of minute features. Serial number restoration are forensic scientific methods to recover a manufacturer's identifier that has been erased. Fingerprint analysis looks for fingerprints on weapons and cartridges. What can be used as evidence? Gunpowder residues can help identify the shooter or the location where a firearm was used. Termination marks can help identify the location or trajectory of the projectile. Spent bullet cartridge cases can do like and subscribe. A document examination unit in forensics refers to a specialized section within a forensic laboratory that examines disputed documents, like handwriting samples, signatures, printed materials, or any other document where authenticity or origin is questioned, to identify potential alterations, forgeries, or inconsistencies, and provide expert analysis for legal proceedings. Essentially, they analyze questioned documents using scientific methods to determine their validity and source. Key points about Document Examination Unit. Focus on questioned document. The primary function is to examine documents where there is doubt about their authenticity, authorship, or alteration, including signatures, wills, contracts, checks, ransom notes, etc. Expert analysis. Trained forensic document examiners compare characteristics of the questioned document to known samples, like a suspect's handwriting, to determine if they match or not. Techniques used. Handwriting analysis. Examining the features of handwriting, spacing, line quality, and pressure. Ink analysis. Studying the chemical composition of inks to identify different ink sources or potential alteration. Paper analysis. Examining the paper type, watermarks, and manufacturing detail. Indented writing. Analyzing impressions left on a page from writing pressure. Electrostatic detection apparatus. ESDA, a non-destructive method to visualize indented writing. Microscopy. Using microscopes to examine minute details on the document. Court testimony. Forensic document examiners often provide expert testimony in court regarding their findings on questioned document. Forensic photography is a systematic process that uses photography to document crime scenes and physical evidence. It's an important part of the criminal justice system. Its purpose is to record the appearance of physical evidence as it was found, to provide investigators with a photographic record of the scene, to present the crime scene in court for judges and counsel. Techniques used in photography include crime scene preservation. Prioritize preserving the crime scene before taking photograph. Lighting and angles. Proper lighting and angles are important for the quality of forensic photograph. High dynamic range. HDR photography combines multiple images taken to create more detail. Exposure. Exposure meters measure the amount of light falling on a subject. Types of photographs: overall, mid-range, and close-up. Role in investigation. Forensic photography is involved in almost all sections of a forensic science lab. It's also important for examining engine and chassis numbers of suspected vehicle.